Hey guys, I am Akanksha. Welcome back to my channel Akanksha's Vision. Today we are going to discuss about the scientist in molecular basis of inheritance chapter. Don't miss this video also, as this is also an important chapter in 12th biology, as well as I will be covering this chapter in two parts. So let's start with today's part, and don't go anywhere, and don't forget to like, subscribe and share as much as possible. Our first scientist is Frederick Griffith, who in 1869 identified DNA as an acidic substance, plus he termed DNA as nuclein. Our next scientists are James Watson and Francis Crick. We all have heard about them very well as I have mentioned about their work and contributions in my previous videos. So guys, do check them out. And let's talk about what they did in 1953. They proposed the double helical model for the structure of DNA in 1953 on the basis of the X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin. Erwin Chagaf was the one who proposed Chagaf Rule. Chagaf Rule states that the ratio for the double-stranded DNA between the adenine and thymine and guanine and cytosine is constant and equals 1. Now guys, there is one thing to remember in Chagaf Rule, which is that it does not apply on the single-stranded nucleic acids. Till now we have discussed about the general introduction, that what was DNA, how DNA was discovered, who gave the structure of DNA. Now guys, now let's understand that what was the genetic material. Was it DNA or protein or RNA? Let's see. So guys, let's start with Frederick Griffith's experiment, which basically focused on transforming principle. In 1928, Frederick Griffith performed a series of experiments with Streptococcus pneumoniae and witnessed a miraculous transformation in the bacteria. Basically, he has taken two strains of the bacteria. First is S strain and second is R strain. S strain had mucus or the polysaccharide coat, whereas R strain did not. Therefore, S strain was found to be virulent. So guys, he basically did 4 attempts, in which in the first attempt he has taken S strain and injected into the mice, and mice was found to be dead. Second attempt, R strain was injected into the mice and mice was found to be alive. In the third attempt, he injected S strain to be heat killed into the mice, and mice was found to be alive. And lastly, he injected S strain heat killed along with R strain, which was alive, into the mouse, and therefore it was found that mice was dead. So he concluded that there was something that transferred from the S strain to R strain that basically changed the nature of R strain to be virulent. So he gave it as the name of transforming principle, and that this could be possible only when the genetic material was transferred. So यहाँ से अब जो बायोकेमिकल नेचर है जेनेटिक मटेरियल का, वो पता लगाने की शुरुआत हुई. From 1933 to 1944, 3 scientists, Oswald Avery, Macklin McCarty and Colin MacLeod, they worked to determine the biochemical nature of the transforming principle in Griffith's experiment. They discovered that protein digesting enzymes, proteases, and RNA digesting enzymes, RNases, did not affect transformation, so the transforming substance was not a protein or RNA. Digestion with DNAs did inhibit transformation, suggesting that DNA caused the transformation. Therefore, they concluded that DNA is the hereditary material. Guys, not every biologist was convinced by this experiment. So, the unequivocal proof that the DNA is the genetic material came from the experiments of Alfred Hershey and Martha Chase in 1952. Hershey and Chase worked with bacteriophages as well as bacterial cells. Bacteriophages are basically the viruses that attacks the bacteria. Now guys, let's understand why they have taken the bacteriophage as well as bacteria in their experiments. Whenever bacteriophage attacks bacterial cell, what bacterial cells do? Bacterial cells just manufactures the genetic material of bacteriophage, understanding that it is its own. Therefore, Hershey and Chase worked on them to identify whether it was protein or DNA that passed from virus to bacteria. What they did in their experiment was that they grew some viruses on a medium that contained radioactive phosphorus and some others on a medium that contained radioactive sulfur. Viruses grown in the presence of radioactive phosphorus contained radioactive DNA but not radioactive protein, because DNA contains phosphorus but protein does not. Similarly, viruses grown on radioactive sulfur contain radioactive protein but not radioactive DNA, because DNA does not contain sulfur. This was how the search for the genetic material was over. Now let's talk about the semi-conservative nature of DNA replication. Matthew Michelson and Franklin Steele, they experimentally proved that DNA replicated semi-conservatively. They did experiments in 1958. Now let's talk about their steps involved in their experiment. पहला स्टेप में दे ग्रू ई कोलाई इन अ मीडियम कंटेनिंग NH4Cl. यहाँ पे जो NH4Cl में नाइट्रोजन का जो आइसोटोप यूज़ किया गया था वो इट वॉज N15 आइसोटोप for many generations, and the result was that N15 was incorporated into newly synthesized DNA. Now, the difference between N15 isotope and N14 isotope was that कि जो N15 isotope है वो heavy isotope था नाइट्रोजन का. Therefore, जो heavy DNA molecule था वो easily distinguish किया जा सकता था from the normal DNA molecule just by doing centrifugation in cesium chloride density gradient. They transferred these cells into a medium with normal NH4Cl, जिसमें नाइट्रोजन का N14 आइसोटोप था rather than N15 आइसोटोप, and took samples at various definite time intervals as the cells multiplied and extracted the DNA that remained as double-stranded helices. They concluded that the DNA that was extracted from the culture one generation after the transfer from N15 to N14 medium had a hybrid or intermediate density. Now guys, in simple language, let's see the timeline from 0 minute. जब स्टार्ट किया था एक्सपेरिमेंट तब हमने लिया था तो हमारे पास आफ्टर 20 मिनट्स जो DNA मिला दैट वाज 100% हाइब्रिड. देन आफ्टर 40 मिनट्स स्टार्ट फ्रॉम द स्टार्टिंग पॉइंट ऑफ द टाइमलाइन the DNA was 50% hybrid, 50% unhybrid. And similar experiments was performed involving the use of radioactive thymidine to detect the distribution of newly synthesized DNA in the chromosome was performed on Vishya Faba Beans by Taylor and colleagues in 1958. So guys, for today this was all from my side. Let's meet in next video. I hope my videos must be helping you out, and don't forget to like, subscribe and share. All the best guys. Happy learning. Thank you for watching.